Module 1, Protein Coding Genes, Structure and Expression. The learning objectives for this module are to describe gene organization and structure, including exon and introns, define an open reading frame, to explain the splicing process and its significance in conveying DNA information, to illustrate the process of cellular protein synthesis, including post-translational modification and folding, and finally to distinguish the various levels of protein structure. Scope of our genome. Our genome is complex, comprising about 3.2 gigabases, or 3.2 times 10 to the ninth base pairs of DNA. One of its main tasks is to produce a huge variety of different proteins that dictate how cells work. Surprisingly, however, coding DNA that specifies the polypeptides of our proteins, accounts for just over 1.2% of our genome. The remainder of our genome is non-coding DNA that does not make any protein, but a significant fraction of the non-coding DNA is functionally important. Functional non-coding DNA sequences include many different classes of DNA regulatory sequences that control how our genes work, such as promoters and enhancers, and DNA sequences that specify regulatory elements that work at the RNA level. We now know that in addition to our protein coding genes, which make a messenger RNA that makes a polypeptide, we have many thousands of genes that instead make different classes of functional non-coding RNA. Some of these RNA genes, such as the genes encoding ribosomal RNA and transfer RNA, have been known for decades. But one of the big surprises in recent years has been the sheer number and variety of non-coding RNAs in our cells. In addition to the RNA genes, our protein coding genes frequently make non-coding RNA transcripts as well as messenger RNAs. These include both linear and circular RNAs, and many of these RNAs have important regulatory roles. Like other complex genomes, our genome has a large proportion of moderate to highly repetitive DNA sequences. Some of these are important in centromere and telomere function, while others are important in shaping how our genome evolves. Protein organization, exons and introns. The protein coding genes of bacteria are small, on average about 1,000 base pairs, and are quite simple in their organization. Eukaryotic genes, however, are larger and more complex. Most eukaryotic protein genes are split into segments called exons that are separated by non-coding DNA sequences called introns. The number of exons and introns varies considerably between genes, and there seems to be little logic about precisely where introns insert within genes. Excluding single exon genes, that is, genes that lack introns, average exon lengths show moderate variation from gene to gene. However, in complex genomes, such as those of the human genome, introns show extraordinary size differences. Our genes are therefore often large and sometimes extending over more than a megabase of DNA. The table shows examples of differential gene organization. In some cases, we see that a gene is comprised of a single exon and no introns, as in the case of SRY gene, which is found on the male chromosome, the Y chromosome, and plays a role in sex determination. However, we have some genes that are made up of numerous exons. The DMD, or the dystrophin gene, is made up of 79 exons, and the average size of each exon is 180 base pairs. And the number of introns is quite large, with the average size of 30,770 base pairs. The entire size of this particular gene is 2,400 kilobases. So we have a very wide range, and then of course there are things that fall in between, but you can see there's quite a wide variety in complexity. The transcriptional unit. So again, in thinking about the topic of this module, that is the production of proteins from RNA, one must recall that, like all genes that are split into exons are initially transcribed by an RNA polymerase to give a long RNA transcript. And this is sometimes referred to as the primary RNA transcript. And the primary RNA transcript is identical in base sequence to that of the transcribed region of the sense strand of the DNA, except that U replaces T. The transcribed region of DNA is called a transcription unit. Thereafter, the primary RNA transcript undergoes a series of processing events that give rise ultimately to the mature RNA transcript. So keeping in mind this process of transcription, whereby we take that information from the DNA strand and produce the primary transcript, which is homologous to the sense strand of the DNA. It's produced by the RNA polymerase. It's a process that takes place in the nucleus. It's homologous to the sense strand, but we find U instead of T. And then ultimately, this RNA needs to undergo a series of modification, the first and perhaps most important of which is RNA splicing. And in the process of RNA splicing, we remove the introns that divide up the exons of the particular gene. RNA splicing is the process of intron removal to generate the mature RNA transcript. As widespread as RNA splicing is amongst complex genomes, approximately 10% of human protein coding genes do not undergo RNA splicing. And so that means that these genes, or the genes that encode histone proteins, are comprised of a single exon. Splicing gives a great deal of flexibility, plasticity to our genome, in that we can create a variety of gene products through the process of alternative splicing. So alternative splicing refers to the fact that one RNA transcript can be spliced differentially depending on the particular signals that are present, to give rise to a variety of gene products. So one gene can give rise to several different proteins depending on how the RNA is spliced. Alternative splicing patterns, then, a single gene can give rise to several different gene products that can be actually functionally diverse. Alternative splicing is important in addition because it aids in genome variation. Now, when we consider splicing, so we have picture in your mind this primary RNA transcript, and there are particular DNA sequences that are known as splice sites, and these are the sites that direct the splicing machinery to cleave and then rejoin the DNA fragments once the introns have been removed. And these are called splice sites. And splice sites are very important in genetic disease because if we alter where the splicing occurs, that can lead, as we'll see later in this talk, to a non-functional gene product and therefore a disease state. There are certain types of sequences. GU, for example, is characteristic of a splice donor site, and AU is a splice acceptor site. If we're thinking about the concept of splicing in and then splicing out. How does splicing take place? Splicing takes place in part through groups of protein and RNA factors called spliceosomes. And so the spliceosomes actually carry out the process of RNA splicing. It depicts the process of RNA splicing. Most of our protein coding genes and many RNA genes undergo RNA splicing, as we have described earlier. In this generalized example, a protein coding gene is illustrated with an upstream promoter and 3 exons. And those are shown in blue, that are separated by 2 introns that each begin with the dinucleotide GT and end in the dinucleotide AG. You can see that they begin with an AG and end with a GT. The central exon, exon 2, is composed entirely of coding DNA, but exons 1 and 3 have non-coding sequences that will eventually be used to make untranslated sequences in the mRNA. The 3 exons and the 2 separating introns are transcribed together to give a large primary transcript. The RNA transcript is cleaved at positions corresponding to the exon-intron boundaries. The 2 transcribed intron sequences that are excised are each degraded in the cell, but the transcribed exon sequences are joined or spliced together to form a continuous mature RNA that has non-coding sequences at both the 5' and the 3' ends, and these are depicted as 5' UTR for untranslated units and 3' UTR for 3' untranslated units or regions. The central coding sequence of the mRNA is defined by a translation start site, which is almost always the trinucleotide AUG. So when you see AUG within an exon, you know that that is where the start site of translation is going to occur. And a translation stop site, which is at the end of the coding region, and is read or translated to produce a polypeptide in the 5' to 3' direction. Translation. Messenger RNA, mRNA molecules are produced by RNA splicing in the nucleus and then ultimately are exported to the cytoplasm. Here they are bound by the ribosomes, very large complexes consisting of 4 types of ribosomal RNAs and many different proteins. Key features of translation are that it is a cytoplasmic process. It involves the RNA molecules tRNA, the ribosomal RNA molecules, and ribosomes. It produces a protein using the mature mRNA as a template. And the purpose of translation is to translate the codons, the units that are made up of 3 nucleotides to produce this polypeptide from the mRNA template. And just a couple things to recall about the codons. During the process of translation, the mRNA is read or translated in groups of 3 nucleotides at a time, and these 3 nucleotides are the codons. And so there is a correspondence between 3 nucleotides and each of the 20 amino acids that we have. And if we really think about it, there are actually 64 possible codons, 4 possible bases at each of the 3 nucleotide positions. So that's 4 times 4 times 4. And of these 61 of them are used to specify a given amino acid. And the 3 others constitute the stop codons, the signal the end of protein synthesis. And the genetic code is universal, so that is used in all organisms, whether we're talking about E. coli or C. elegans or mice or man or plant. That genetic code is universal. And the genetic code is really the set of rules that dictate how codons are interpreted. And do recall again that the genetic code has some redundancy. That means that there can be a particular codon that can specify the same amino acid. Consider serine, for example, and serine can be specified by any of the 6 codons, which include UCA, UCC, UCG, UCU, AGU, and AGG. On average, an amino acid is specified by any of 3 codons. And frequently that last position in the codon is the one that has the greatest range of flexibility. Consider the product of translation, and that is the polypeptide. And so let's take a look at the polypeptide and amino acid structures. In this particular slide, we show the polypeptide primary structure. That's the simplest level of structure, and that really is the sequence of amino acids. And here we have 1, 2, 3, 4, 5 amino acids. So a pentapeptide is shown here, and with its 5 amino acids highlighted. And here we have the left endmost of the polypeptide. So whenever we read the sequence of a polypeptide, we read it from the endmost terminus left to right. And so we notice that the polypeptide begins with an amino group and ends in a carboxyl group. So it has an N terminus and a carboxy terminus. The side chains are, as depicted in this particular diagram, give the chemical specificity or the biochemical specificity to the particular polypeptide chain, and we'll show in the next slide how these R groups can be made. You will notice the type of bond that is joining. This is an amide bond, and it joins adjacent amino acids together in the polypeptide chain. Down here is the neighboring amino acids joined together in the peptide bond. So this is the specific type of amide bond that is formed between the carboxyl group of one amino acid and the amino end of the other amino acid. Peptide bond is formed by condensation reaction between the carboxyl group of one amino acid and the end amino group of another amino acid. Very stable type of covalent bond. Now shown in this slide are the 20 different amino acids, the principal amino acids that we find in proteins. And as depicted are their side chains. So you can see in the amino acid glycine, there really is no side chain functional group. We have a hydrogen atom. Whereas in alanine, we have a methyl group, and then it gets increasingly complex in variability. So we have several hydrophobic amino acids, and that would be alanine, valine, leucine, isoleucine. So these are all very similar in composition, and if you recall back to the terminology we used earlier about missense mutations, this would be considered a valine for leucine would be considered a conservative mutation because they're in the same chemical family as R groups. And then we have the serine and the threonine amino acids, and these are both polar amino acids where the R group involves a hydroxyl group. This is significant because these hydroxyl groups can be acted upon and different entities can attach on to this R group through the, for example, they can be phosphorylated through this hydroxyl group, but serine and threonine comprise another group of amino acids. Then we have the acidic amino acids, aspartic acid and glutamic acid. These have another carboxyl group, as you see in this particular instance. And you'll notice another thing to note in this diagram is that we have a spelling out of the name, but we also have two types of abbreviations for the amino acids. Historically, amino acids were designated by three-letter abbreviations, so SER for serine, for example, and then we also have a single letter nomenclature as well for the amino acids. Some of these are more intuitive than others. So for example, glycine is denoted by G, the letter G, whereas tryptophan is denoted by the letter W. And so as we carry on, we have polar amino acids, asparagine and glutamine, basic amino acids, lysine, arginine, and histidine. Histidine, you'll note, has a cyclic ring. These amino acids are positively charged. The side chains are positively charged at physiological pH. And then we have another amino acid on the last row, cysteine. Cysteine is unique in that it contains a sulfhydryl group, which is reactive. And this can be important in the stability and function of the protein that contains a cysteine residue. Methionine also contains a sulfur group. And then we go to the amino acids that have aromatic side chains, and this includes phenylalanine, tyrosine, and tryptophan. Tyrosine is somewhat unique in that it is a aromatic side chain, but it also contains a hydroxyl group. And then last but not least, we have proline, which is a wholly cyclic amino acid in the sense that the N-terminus is part of the cyclic ring. So proline is a very unique amino acid, and it has some very significant ramifications in the structure of a protein due to its ring-like structure. Translation. Translation begins when ribosomes bind to the 5' end of mRNA and then move along the RNA to find a translational start. Translation is comprised of three phases: initiation, elongation, and termination. It involves the mature mRNA, and if we think about the mature mRNA, it actually has three regions: the central coding region, and then in addition to that, 3' and 5' untranslated regions. In initiation, certain sequences are important, and that includes the Kozak sequence. The process of translation begins when the 5' end of the mRNA moves along to find the start site, or the initiation codon, which is an AUG trinucleotide embedded within the broader, less defined Kozak sequence. So you can see this Kozak sequence here is shown, GCC, and then a purine nucleotide, and then CCAUG, which is the actual start codon, and the nucleotides that are shown in bold are the ones that are more highly conserved. So nearly all proteins then, because of AUG codes for a methionine, if you'll recall back to our previous slide, nearly all amino acids begin with methionine. The initiation codon is the start of an open reading frame. That is the start of the amino acids in the polypeptide. And the process of translation terminates when the ribosome encounters a stop codon. So within the mRNA sequence, once the ribosome encounters one of three types of codons, either a UAA, a UAG, or UGA, then the last amino acid will be incorporated at that particular location. Slide depicts the basics of translation pictorially. In A, we see a ribosome attaches to the 5' untranslated region, or the 5' UTR of the mRNA, and then slides along until in B, it encounters the initiation codon, AUG, at which point a methionine-bearing transfer RNA, which is not shown, engages with the AUG codon and deposits its methionine, which is shown by a green bar. In C, we see the ribosome continues to move along the mRNA as it encounters each codon. In turn, a specific amino acid bearing tRNA is recruited to recognize the codon and to deposit its amino acid according to the genetic code. The ribosome itself is catalyzing the formation of a peptide bond between each new amino acid and the last amino acid, forming a polypeptide chain, which is shown here as a series of joined green bars. Then in D, finally the ribosome encounters a stop codon, at which point, as shown in E, the ribosome falls off the mRNA, dissociates into its two subunits, and releases the completed polypeptide. As discussed in subsequent slides, the polypeptide will then undergo post-translational modification to produce the mature protein. Shown in this slide are the transfer RNA and its structure, and its description as its role as an adapter RNA in the translational process. On the left-hand side of this slide, shown in A, we see transfer RNA structure. The tRNA GLY, so the tRNA for the amino acid glycine, is shown here and illustrates the classic cloverleaf tRNA structure. So you see it does appear somewhat like a cloverleaf, and this is formed through intermolecular base pairing, produces this 3-arm terminating loop plus the acceptor arm that binds the amino acids through the covalent bonding of the 3' end. So if you look at the 3' end, this is where the particular amino acid, in this case glycine, will be carried by this tRNA molecule. The 3 nucleotides at the center of the middle loop form what is known as the anticodon. So the anticodon is going to recognize the codon on the corresponding mRNA molecule, and it is the tRNA, this part of the tRNA, that will really specify the docking to the codon. You'll notice that tRNA molecules contain unusual nucleotides, for example, the 5,6-dihydrouridine, pseudouridine, methylcytidine, methyladenosine, and O-methyluridine. So, there's some very important parts of the tRNA molecule, in particular the 3' end, which gets charged with a particular amino acid. That is where at that 3' end the amino acid will be attached, and then at the base we have the anticodon, which will dock with the specific codon. And then in B, we show the role of the adapter RNA. So different tRNAs carry different amino acids according to the type of anticodon that they bear. So as a ribosome traverses an mRNA, it identifies the AUG codon, and a tRNA molecule that bears a methionine and the complementary anticodon sequence CAU. So the AUG will base pair with CAU and engages the ribosome so that the CAU anticodon will then base pair, and for ease of illustration, it's shown that the tRNA in the opposite left orientation to the standard form shown in A. Therefore, a tRNA bearing glycine engages the second codon, GGG, after base pairing with its CCC anticodon. The ribosome's peptidyltransferase then forms a peptide bond between the N-terminal methionine and the glycine. So key features to note on here is that the ribosomal RNA contains a peptidyltransferase activity that forms the peptide bond. So the peptide bond is not being formed through an enzyme, but rather a ribosome. And this process continues as we see the next tRNA molecule will come in and dock and join the serine molecule to the glycine molecule through the ribosome RNA peptidyltransferase activity. This slide depicts the concept of the reading frame and its significance. So during translation, the way in which the continuous sequence of the mRNA is read as a series of triplet codons is referred to as the reading frame. There are three possible forward reading frames for any mRNA, and the correct reading frame is set by the correct recognition of the AUG initiation codon. In this particular slide, we use 3-letter words to illustrate how meaning can be altered by changing the transcriptional reading frame. The sequence of letters at the top, reading frame 1, reading frame 2, reading frame 3, so the sequence of letters at the top can be grouped into sets of 3 codons that make sense in reading frame 1. The old man got off the bus and saw the big red dog and her pup, but make no sense when using reading frame 2 or reading frame 3. Okay, so the sequence up at the top of the rows, which is in black, could be read in 3 different ways, and depending on when you start, where you make the cut into triplets really affects the logic of this. So as we can see in figure 1, the reading frame 1 makes sense, but making a shift to reading frame 2 or 3 produces nonsense. The same principle generally applies to coding sequences. So, for example, if 1 or 2 nucleotides are deleted from a coding sequence, in the case of a mutation, the effect is to produce a frame shift, a change of the reading frame that will result in a nonsense translation. So this helps to reinforce that notion. This particular slide shows how the splitting of codon sequences can be affected by introns. At the DNA level, introns may interrupt a codon sequence at one of 3 types of a position: a point precisely between 2 codons, between the first and second nucleotides of a codon, or between the second and third nucleotide of a codon. An internal exon may be flanked by introns that insert at the same type of position. And an exon like this, sometimes called a symmetric exon, the number of nucleotides is always exactly divisible by 3. An internal asymmetric exon has a total number of nucleotides that is not exactly divisible by 3, and so is flanked by introns that insert at different types of positions. In this figure, we imagine that the same sequence is split by 2 introns, 1 and 2, shown here in green and in orange. The green introns both interrupt the coding sequence between codons, and so the sectional exon processing is symmetric. Following release of the newly synthesized polypeptide to produce a mature protein requires several steps, 1 and 2. Polypeptide typically undergoes some type of post-translational proteolytic cleavage. In this case, chemical modification is symmetric. Proteins also need to fold
Mutations
properly
that cause
and
an
often
internal exon to be
bind
deleted
to other
or not
polypeptides
included in
forming
splicing
part
have
of a
less of an effect
multi-subunit
in the
protein.
case of symmetric exons.
And then there is the
Loss
need to
of the
transport
symmetric
the
exon
protein to
2
the correct
in figure
intracellular or
2b
extracellular
leads to
location.
the loss of 5 words or codons. But
This table shows the common types of chemical modification of proteins by covalent addition of chemical groups to a side chain. Sometimes small chemical groups are attached to the side chains of specific amino acids. These groups can sometimes be particularly important in the structure of a protein, as in the case of collagens, which have high levels of hydroxyproline and hydroxyserine. In other cases, dedicated enzymes add or remove small chemical groups to act as switches that convert a protein from one functional state to another. Thus, specific kinases can add a phosphate group that can be removed by a dedicated phosphatase. Yet, in other cases, proteins can be modified by attaching complex carbohydrates or lipids in a polypeptide backbone. For example, proteins that are secreted from cells or that are destined to be part of the excretory processes of cells are routinely modified so as to have oligosaccharides covalently attached to one side of chains of specific amino acids. Different types of lipids are often added to proteins that function within the lipid bilayer of a membrane. Folding. The amino acid sequence, the primary structure, dictates the protein patterns of folding, and specific regions of the polypeptides can adopt specific types of secondary structure that are important in how a protein folds. Until correct folding has been achieved, the protein is unstable. Different chaperone molecules help with the folding process. Careful supervision is needed because partially folded or misfolded proteins can be toxic to cells, as in the case of Alzheimer's disease. For many proteins, notably globular proteins, the folding pattern is stabilized by a form of covalent cross-linking that can occur between certain distantly located cysteine residues. The thiol groups of the cysteine residues interact to form a disulfide bond. Shown in this slide are the elements of protein secondary structure. On the left in A, we see an alpha helix. The solid rod-like structure is stabilized by hydrogen bonds between the oxygen of the carboxyl group of each peptide bond and the hydrogen on the peptide bond of the fourth amino acid away, yielding 3.6 amino acids per turn of the alpha helix. The side chains of each amino acid are located on the outside of the helix. There's almost no free space between the helix. Note, only the backbone of the polypeptide is shown, and some of the bonds have been omitted for clarity. In B, a second form of secondary structure is shown. A beta sheet, also called a beta pleated sheet, is shown here. The hydrogen bonding in this case occurs between the carbonyl oxygens and amide hydrogens on adjacent segments of a sheet that may be composed of either partial segments of the polypeptide as shown here, or antiparallel segments that travel from the N terminus to the C terminus. So we essentially have parallel or antiparallel beta sheets, and these are stabilized by hydrogen bonds, but between the adjacent rows of amino acids, as opposed to in the helical structure that we saw in A. Intra-chain and inter-chain disulfide bridges in human insulin. We're going to use human insulin as an example of how disulfide bonds are involved in types of protein structure. So human insulin is composed of two polypeptide chains, the A chain that contains 21 amino acids and the B chain, with 30 amino acids. Disulfide bridges or disulfide bonds, that is a covalent bond between two sulfhydryl groups on adjacent chains, form by a condensation reaction between the sulfhydryl groups on the side chains of the cysteine residues. They form between the side chains of cysteines at position 6 and 11 within the insulin A chain, and also between the cysteine side chains on the insulin A and B chains. Note that here all the cysteines participate in disulfide bonding, which is unusual. When disulfide bonding occurs in large proteins, typically only certain cysteine residues are involved. Cleavage and transport. The initial polypeptide normally undergoes some type of N-terminal cleavage. This often involves removing just the N-terminal methionine. The AUG initiation codon specifies methionine, but the amino acid sequences of mature polypeptides rarely begin with methionine. In other cases, the N-terminal cleavage can be more extensive. For proteins that are secreted from cells, the polypeptide precursor carries an N-terminal leader sequence, sometimes referred to as the signal peptide, that is required to assist the protein to cross the plasma membrane, after which the signal peptide is cleaved at the membrane, releasing the mature protein. The signal peptide, which is often 10 to 30 amino acids in length, carries multiple hydrophobic amino acids. And lastly, there are several proteins, for example insulin, which are synthesized initially as preproteins and subsequently undergo a series of proteolytic cleavages to yield the final mature polypeptide. And this concludes Module 1.